Now we'll move to our next speaker, Dr. Kalesh Karunakaran. So he is an MSc in chemistry from IIT Madras and Ph.D. in chemical biology from National University of Singapore. Following Ph.D., he was a senior research scientist at AMRI Singapore during 2010 to 2012 and then a Marie Curie Fellow from 2012 to 14 at Imperial College London. He was an MRC Post-Doctoral Fellow from 2014 to 15 and GSK-Imperial College London Engineered Medicine Laboratory Post-Doctoral Fellow from 2015 to 18 before beginning his independent career at Durham University UK in 2019 as an assistant professor in research. His research lies at the interface between synthetic chemistry and the life sciences in the field of chemical biology and chemical proteomics. His current research, supported by the MRC-Global Challenges Research Fund, focuses on the development and application of creative chemical tools and technologies to understand and manipulate living systems, with an emphasis on processes important to human diseases. With this, I request Dr. Kalesh to give his talk on chemical proteomics to unravel drug targets, protein function, and post-translational modifications. Over to you, Dr. Kalesh.
Good afternoon, everyone. First of all, many thanks to Professor Jadhav for the invitation to give a talk in this virtual conference on drug discovery. Deconvoluting the mode of action of drugs or developmental compounds remains one of the biggest challenges in drug discovery today. As a matter of fact, only a tiny fraction of the advanced stage developmental compounds make it to the market, causing substantial economic burden in the pharmaceutical sector. So the billion-dollar question is, how can we mitigate this problem? Well, there is no simple solution to this problem, but over the last several years, we have realized that the main problem is our lack of understanding of how drugs work in a biological system, which is highly dynamic and complex. Our age-old concept of one drug engaging with one biological target is responsible for the effect that you see. Therefore, by blocking that target, you can develop a successful drug, is an extremely oversimplified view. So in order to tackle the problem, we need to have innovative tools and technologies that can address the complexity of the biological system. Chemical proteomics is one such approach where we use creative chemical tools to understand the living systems. It is a multidisciplinary approach that involves synthetic chemistry, biochemistry, cell biology, mass spectrometry-based proteomics, and substantial computational and statistical approaches. Today, I'm going to talk about some of our recent research in chemical proteomics that we conducted at Durham University as well as at Imperial College London. The vast majority of drug targets are proteins as compared to other biomolecules. Proteins are the workhorses of the cell. But proteins are highly dynamic, and some of them undergo covalent chemical modifications, which is completely beyond any sort of genetic control. So this is an additional level of complexity that no genetic method that you use in drug discovery can address. Most of these modifications occur post-translationally, so are called post-translational modifications, or PTMs in short. Most PTMs occur enzymatically. For instance, protein phosphorylation is catalyzed by protein kinases. PTMs can be reversible or irreversible. Phosphorylation is an example of a reversible PTM. It is reversed by protein phosphatases, for instance. PTMs, irrespective of whether they are reversible or not, make decisive roles in cellular functions. For instance, a PTM may change the enzymatic activity of the substrate. It may switch the protein binding partners or alter the subcellular location of the protein. So PTMs provide a combinatorial control of protein function in space and time. There are over 200 PTMs known today. What is most important is that abnormalities in PTM regulation often leads to complex human diseases, including cancer and degenerative diseases. PTMs are also targets in infectious diseases. So now the question is, how do we study PTMs? There is an estimate of about 10 billion proteins in every cell. But at any given point of time, only a tiny fraction of the cellular proteome will carry any particular PTM of your interest. Therefore, direct investigation of any PTM in cells is highly challenging because now you are working against a large background of other cellular proteins. So most PTMs need to be enriched prior to studying them. The scenario would be a lot simpler if we can install a selective chemical handle onto the PTM of our interest so that we can use that handle to fish out the modified proteins from the pool of other cellular proteins. If that is possible, let's say, then we can address important questions like which proteins are modified, where are they modified, which enzymes modify them, what is their importance in disease, can we target them for intervention, etc. Now, in part one of this presentation, I'm going to talk about an important post-translational modification called ADP-ribosylation, which is currently being targeted in cancer. Our approaches to understand and characterize this important protein modification will be covered. Okay. I would call ADP-ribosylation an elusive protein modification. Arguably, it is one of the most difficult post-translational modifications to study because of various reasons. Number one, its structural complexity. First of all, you can have either mono ADP ribosylation, where only a single unit of ADP ribose is added to the target protein. And the second version, which is more well known, is the poly ADP ribosylation, which is highly complex and a heterogeneous modification. As you can see here, the modification with so many branches. The polymeric PTM may contain up to 200 monomeric units. In addition to this structural complexity, the fact that the modification occurs on a large variety of amino acids make it even more complex. It is difficult to study. There are no specific and sensitive antibodies available, and the available chemical-based enrichment methods are all non-specific. Therefore, ADP ribosylation, despite its importance in cancer, remains a poorly characterized post-translational modification. ADP ribosylation is catalyzed by a family of proteins known as poly-ADP ribosyl polymerases, or PARPs in short form. We have got 16 different PARPs in humans. The majority of them are only mono ADP ribosylating. The majority of PARPs are poorly characterized, as mentioned earlier, and we don't know much about their substrates or their functional roles in cells. The only conclusive knowledge that we have is that PARP1, the founding member of PARPs, is directly involved in DNA damage response. And PARP1 inhibitors are currently being developed as anti-cancer agents. We have been working on ADP ribosylation since 2015 on a GSK-funded project. While we were focused on developing mass spectrometry methods, a paper appeared in Nature Chemical Biology from a US group. They reported that 6-alkyne adenosine, which is an adenosine derivative with a terminal alkyne functionality at position number 6 of the adenine, when treated with cancer cells, will get uptaken, and once inside the cells, the NAD synthesizing enzymes will utilize this as a starting material for making NAD, or nicotinamide adenine dinucleotide, the ADP ribose donor of PARP enzymes. Instead of making the natural NAD, in this case, the cells will make NAD analog with an alkyne tag at position number 6. So when PARPs utilize this modified NAD as the ADP ribose donor, the end result is that every single substrate protein of the PARP in the live cell will now contain the alkyne tag in the PTM. Now we can utilize this alkyne tag for selectively fishing out the substrates from the larger pool of other cellular proteins. But around the same time, other reports indicated that position number 2 of the adenine ring in the nicotinamide adenine dinucleotide is more tolerant position for substitutions for PARP recognition. Therefore, we synthesized this compound called 2-alkyne adenosine and tested that for metabolic incorporation. As you can see here in the ingel fluorescence scans, the two compounds more or less performed identically in terms of the protein labeling profiles. Now, for a head-to-head comparison of the labeling performances of the two compounds, we decided to employ a very powerful and highly sensitive quantitative proteomics mass spectrometry method known as TMT labeling or Tandem Mass Tag labeling. The method allows multiplexing of samples for mass spectrometry analysis. It uses isotopically labeled reporter tags that react with amino acid groups in tryptic peptides. Each experimental sample is labeled with a unique isobaric tag, and then the samples are all pooled together and analyzed in a single LC-MS run. In the MS2, fragmentation of the reporter units occur, and by quantifying the relative intensities of the reporter ions from the different experimental samples, we can get a relative quantification of the proteins from those different experimental samples. We performed a head-to-head comparison of the 6-alkane adenosine and 2-alkane adenosine labeling using TMT6 plus quantitative proteomics MS. The analysis of the tandem mass spectra led to robust identification of over 1,600 protein targets in a breast cancer cell line. Intriguingly, a comparison of the labeling performance of the two adenosine analogs reveal that although the protein targets are more or less identical, there are differences in terms of preferences of individual substrates for the cofactor analogs. And as you can see here, the 2-alkane adenosine is more preferred one for a larger proportion of the substrate proteins. For some proteins, for example, for C1QBP here, the 6-alkane adenosine is more preferred cofactor analog. So in order to get a comprehensive coverage of substrates of ADP-ribosylation, we decided to perform a dual metabolic incorporation approach that involves a simultaneous treatment of the two cofactor analogs in cells. The beauty of this approach is that it can be used for a quantitative comparison of the effect of different PARP inhibitors on the large panel of endogenous PARP substrates in any cancer cell line. As you can see here, we have compared the two clinical PARP inhibitors, olaparib and rucaparib, in MDA-MB231 breast cancer cell line using a TMT 10-plex quantitative proteomics mass spectrometry approach. The dual metabolic incorporation approach combined with the TMT 10-plex quantitative proteomics mass spectrometry enabled us to identify over 2,000 PARP substrates in the breast cancer cell line, which is by far the largest coverage of PARP substrates identified in a single study. More importantly, the method enabled to quantitatively compare the effects of different PARP inhibitor treatments to the large panel of endogenous PARP substrates, providing a highly valuable data set for the mode of action studies of these compounds. With this, I now move on to the second part of this presentation. I will talk about application of quantitative chemical proteomics to target profiling of pharmacologically active compounds. Enzymes that form a major class of drug targets are tightly regulated by post-translational modifications. And it is the activity of enzymes rather than mere expression levels that play more important roles in fundamental cellular physiology and also in pathology. Conventional genomic methods and expression proteomics methods cannot report the activity of enzymes. However, there is a chemical labeling approach known as activity-based protein profiling, originally developed by Ben Cravett at Scripps, that can precisely report on the activity of target enzymes in complex biological systems. The method uses chemical probes known as activity-based probes that covalently react with nucleophilic amino acid residues at or near the active site of target enzymes. If we cannot exploit such interactions, we can still use a slightly different version known as affinity-based probes, which can be cross-linked to their target proteins, again in endogenous systems, using photocross-linking, for instance. The probe labeling is typically coupled with tandem mass spectrometry for protein identification and quantification. This method is now widely applied, not just for the activity-based protein profiling of enzymes, but for the target profiling of pharmacologically active compounds or drugs whose targets are otherwise unknown. Zerumbone, for instance, is a pharmacologically active chemical present in a wild ginger plant. Many molecular targets, as shown here, have been proposed for this compound. However, there is no direct evidence for any of these target engagements. Therefore, we decided to develop a quantitative chemical proteomics method for the profiling of cellular targets of this important phytochemical. Firstly, we synthesized the Zerumbone derivative with a terminal alkyne functionality. The alpha-beta unsaturated ketone functionality in Zerumbone acts as well as in the probe, which is essential for its biological activity, acts as a Michael acceptor center for nucleophilic residues from the target proteins to form a covalent bond. The alkyne functionality, on the other hand, in the probe, will now facilitate fishing out of Zerumbone-bound target proteins from the larger pool of cellular proteins. Now we can perform this whole experiments in the presence of increasing amounts of the parent compound Zerumbone, and in this case, for the real targets, you should see a concentration-dependent decrease in the probe target engagement with increase in concentration of the parent compound. This is called competitive activity-based protein profiling approach. As you can see here, we have performed the labeling experiments in cell lysates as well as in intact cells. The labeling profiles in both cases were significantly different, clearly indicating that in order to identify physiologically relevant targets, we need to perform cell-based experiments rather than experiments using cell extracts. We have then combined the competitive activity-based protein profiling approach with an innovative quantitative proteomics mass spectrometry method known as spiking SILAC. Basically, this involved generating heavy isotope labeled cancer cell line and treatment of the probe compound in a fixed concentration in both the heavy isotope labeled cell line and across batches of the same cancer cell line without any isotope labeling. The probe treated normal cell line were also treated with different concentrations of the parent compound. After click chemistry-based affinity enrichment and tryptic digestion, the samples were all pulled together and analyzed by tandem mass spectrometry. Analysis of the MS1 spectra clearly showed parent compound concentration-dependent decrease in the labeling of real targets, thus robustly identifying the real targets of this important phytochemical. This methodology is now being applied in a number of other studies. Now, moving on to the last part of this presentation, I will show you yet another application of quantitative proteomics mass spectrometry, which is in the study of protein synthesis in a parasitic organism called Leishmania. Leishmania is the causative agent of a neglected tropical disease called leishmaniasis. It is a poverty-related disease. There are two clinical forms of the disease: the cutaneous form, which is manifested as really horrible-looking and highly stigmatizing skin lesions. The second form is a visceral disease. It is also known as Kala Azar in India. It is a deadly disease. If left untreated, the patient will die. Currently available drugs have serious limitations. Better and safer drugs for leishmaniasis is an urgent priority of the developing world. The Leishmania parasite has got an incredible ability to adapt to stressful conditions throughout its life cycle. Understanding the stress adaptation mechanisms of the parasite is of paramount importance because many recent studies indicate that the stress adaptation of the parasite is directly linked to its virulence and infectivity. In order to have a molecular level understanding of the stress adaptation mechanisms of the parasite, we need to first of all identify and characterize the key players involved. The key players involved are obviously proteins. We need to differentially identify and characterize newly synthesized proteins during a stress condition from the pre-existing pool of proteins in the parasite. Now the question is, how do we identify and characterize these newly synthesized proteins in the parasite when it is experiencing a stress? If we can have a chemical handle into every single newly synthesized protein in the parasite when it is undergoing a stress, we can use that chemical handle to fish out those proteins from the larger pool of pre-existing cellular proteins in the parasite. If this is possible, then we can start answering important questions like which proteins are synthesized, how do they respond to a given stress, are there any PTMs in those proteins, how do they interact, can we target these newly synthesized proteins for intervention, or do they have any role in disease, etc. Fortunately, this is possible by a metabolic labeling technology called BONECAT. BONECAT is the abbreviation of bio-orthogonal non-canonical amino acid tagging. In this approach, a methionine surrogate with a click tag, usually acidohomoalanine or homopropagale glycine, is added into a methionine-free cell culture medium. The methionine aminosyl transfer RNA synthetase will recognize this methionine surrogate with more or less similar affinity as that of methionine, so that every single newly synthesized protein will now contain the click tag in it in place of the initiator methionine. This approach has been extensively applied in mammalian systems since its discovery in 2006. It is also reported in bacterial systems and rarely in some parasitic systems. So purely out of curiosity, we decided to find out if BONECAT works in Leishmania. Leishmania parasites were maintained in a methionine-free medium for a short 30 minutes duration. Such a short duration of keeping the parasite in methionine-free medium was found to be sufficient to deplete the intracellular methionine reserves in the parasite so that BONECAT can operate with sufficient sensitivity. After this, acidohomoalanine was added into the culture medium so that the cells can utilize this unnatural amino acid in place of the methionine. After 1 hour, the cells were washed and lysed, and the total cell lysases were subjected to click reaction with a capture reagent containing alkyl moiety and a fluorescent group. Fluorescent imaging of the gel revealed that, yes, BONECAT works efficiently in the Leishmania parasite. Then we asked if this can be used for profiling of newly synthesized proteins in the parasite when it undergoes a stress. The stress that we wanted to study initially was nutrient stress or starvation. The parasites were maintained in PBS for different durations up to 7 hours and performed the BONECAT and compared with the non-starved BONECAT condition. As you can see here in the ingel fluorescence scan image on the left-hand side, the synthesis of the new proteins is significantly downregulated with increase in duration of starvation, which kind of makes sense because the parasite is trying to conserve energy and conserve its nutrients. It doesn't want to waste any energy for translation, which is a highly energy demanding process. But at the same time, you could see that in the early stages of severe starvation, the parasite still synthesizes proteins. So we became curious to know what are these proteins that it synthesizes at this early stage of severe starvation. Because these proteins that it chooses to synthesize even during extreme nutrient stress must be important proteins for stress adaptation in the parasite. Proteins can be identified by a simple mass spec experiment, but we wanted to know precisely to what extent each of these proteins vary relatively between the different durations of starvation compared to a non-starved condition. In order to get that information, we decided to combine the BONECAT approach with an extremely sensitive isotope labeling based quantitative proteomics mass spectrometry technique known as iTRAC labeling. This enabled us to identify hundreds of starvation responsive newly synthesized proteins in the parasite and quantify the relative changes in abundance as a function of duration of starvation. This study provided many useful information for follow-up studies. The beauty of this approach is that the data set that you get at every time point of investigation carries the information on the relative expression of not just one, but hundreds of starvation responsive proteins. And when you compare that with a different time duration, you get the relative changes in abundance of those proteins as a function of duration of starvation. So this data set, for the first time, is enabling us to get a comprehensive and dynamic proteomics picture of how the parasite responds to severe starvation in a laboratory setting. Many follow-up studies based on these findings are currently underway in my group. We have now come to the end of this presentation. The message that I would like to convey is that mass spectrometry-based chemical proteomics is an essential component of modern-day drug discovery. All big pharma companies have now integrated these technologies into their drug discovery platforms. It is important to appreciate that proteins don't work in isolation. Proteins and their signaling pathways form complex network of interactions that often integrate extracellular signals into a complex cell biological response. This protein interaction network is highly dynamic and context dependent. There is a temporal element to it. The message that I'm trying to convey is that in order to develop a holistic understanding of drug action, and in order to save compounds failing in clinical trials, we have to use a discovery proteomics approach and not the conventional approach of studying a protein in isolation. Finally, I would like to thank all my funders and collaborators. My current research is funded by the MRC-Global Challenges Research Fund. The SILAC work was carried out with my Marie Curie Fellowship from the European Commission at Imperial College London with EdPage. The ADP-ribosylation work was carried out with GSK funding, again at Imperial College London during my postdoc with Peter DiMaggio. Patrick Steele, Paul Denny, and John Mina and Ariel Silver are my current collaborators. I'd like to thank you all for listening. Happy to take any questions if you have any.
Thank you, Dr. Karunakaran, for such a wonderful elaborate presentation on application of proteomics in unraveling drug targets.